12. The nervous system and nervous tissue. Figure 12.1. Robotic arms playing foosball. As the neural circuitry of the nervous system has become more fully understood and robotics more sophisticated, it is now possible to integrate technology with the body and restore abilities following traumatic events. At some point in the future, will this type of technology lead to the ability to augment our nervous systems? Credit: U.S. Army, Wikimedia Commons. Introduction. Chapter Objectives. After studying this chapter, you will be able to name the major divisions of the nervous system, both anatomical and functional, describe the functional and structural differences between gray matter and white matter structures, name the parts of the multipolar neuron in order of polarity, list the types of glial cells and assign each to the proper division of the nervous system, along with their functions, distinguish the major functions of the nervous system: sensation, integration, and response. Describe the components of the membrane that establish the resting membrane potential. Describe the changes that occur to the membrane that result in the action potential. Explain the differences between types of graded potentials. Categorize the major neurotransmitters by chemical type and affect the nervous system as a very complex organ system. In Peter D. Kramer's book Listening to Prozac, a pharmaceutical researcher is quoted as saying, "If the human brain were simple enough for us to understand, we would be too simple to understand it." 1994. That quote is from the early 1990s. In the 2 decades since, progress has continued at an amazing rate within the scientific disciplines of neuroscience. It is an interesting conundrum to consider that the complexity of the nervous system may be too complex for it, that is, for us, to completely unravel. But our current level of understanding is probably nowhere close to that limit. One easy way to begin to understand the structure of the nervous system is to start with the large divisions and work through to a more in-depth understanding. In other chapters, the finer details of the nervous system will be explained, but first looking at an overview of the system will allow you to begin to understand how its parts work together. The focus of this chapter is on neural tissue, both its structure and its function. But before you learn about that, you will see a big picture of the system. Actually, a few big pictures. 12.1. Basic structure and function of the nervous system. By the end of this section, you will be able to identify the anatomical and functional divisions of the nervous system, relate the functional and structural differences between gray matter and white matter structures of the nervous system to the structure of neurons, list the basic functions of the nervous system. The picture you have in your mind of the nervous system probably includes the brain, the nervous tissue contained within the cranium, and the spinal cord, the extension of nervous tissue within the vertebral column. That suggests it is made of 2 organs, and you may not even think of the spinal cord as an organ, but the nervous system is a very complex structure. Within the brain, many different and separate regions are responsible for many different and separate functions. It is as if the nervous system is composed of many organs that all look similar and can only be differentiated using tools such as the microscope or electrophysiology. In comparison, it is easy to see that the stomach is different than the esophagus or the liver, so you can imagine the digestive system as a collection of specific organs. The central and peripheral nervous systems. The nervous system can be divided into 2 major regions: the central and peripheral nervous systems. The central nervous system, CNS, is the brain and spinal cord, and the peripheral nervous system, PNS, is everything else. Figure 12.2. The brain is contained within the cranial cavity of the skull, and the spinal cord is contained within the vertebral cavity of the vertebral column. It is a bit of an oversimplification to say that the CNS is what is inside these 2 cavities and the peripheral nervous system is outside of them, but that is one way to start to think about it. In actuality, there are some elements of the peripheral nervous system that are within the cranial or vertebral cavities. The peripheral nervous system is so named because it is on the periphery, meaning beyond the brain and spinal cord. Depending on different aspects of the nervous system, the dividing line between central and peripheral is not necessarily universal. Figure 12.2. Central and peripheral nervous system. The structures of the PNS are referred to as ganglia and nerves, which can be seen as distinct structures. The equivalent structures in the CNS are not obvious from this overall perspective and are best examined in prepared tissue under the microscope. Nervous tissue, present in both the CNS and PNS, contains two basic types of cells, neurons and glial cells. A glial cell is one of a variety of cells that provide a framework of tissue that supports the neurons and their activities. The neuron is the more functionally important of the two, in terms of the communicative function of the nervous system. To describe the functional divisions of the nervous system, it is important to understand the structure of a neuron. Neurons are cells and therefore have a soma, or cell body, but they also have extensions of the cell. Each extension is generally referred to as a process. There is one important process that every neuron has called an axon, which is the fiber that connects a neuron with its target. Another type of process that branches off from the soma is the dendrite. Dendrites are responsible for receiving most of the input from other neurons. Looking at nervous tissue, there are regions that predominantly contain cell bodies and regions that are largely composed of just axons. These two regions within nervous system structures are often referred to as gray matter, the regions with many cell bodies and dendrites, or white matter, the regions with many axons. Figure 12.3 demonstrates the appearance of these regions in the brain and spinal cord. The colors ascribed to these regions are what would be seen in fresh, or unstained, nervous tissue. Gray matter is not necessarily gray. It can be pinkish because of blood content, or even slightly tan, depending on how long the tissue has been preserved. But white matter is white because axons are insulated by a lipid-rich substance called myelin. Lipids can appear as white, fatty material, much like the fat on a raw piece of chicken or beef. Actually, gray matter may have that color ascribed to it because next to the white matter, it is just darker, hence, gray. The distinction between gray matter and white matter is most often applied to central nervous tissue, which has large regions that can be seen with the unaided eye. When looking at peripheral structures, often a microscope is used and the tissue is stained with artificial colors. That is not to say that central nervous tissue cannot be stained and viewed under a microscope, but unstained tissue is most likely from the CNS, for example, a frontal section of the brain or cross-section of the spinal cord. Figure 12.3 Gray matter and white matter of brain removed during an autopsy, with a partial section removed, shows white matter surrounded by gray matter. Gray matter makes up the outer cortex of the brain. Credit. Modification of work by Susino, Wikimedia Commons. Regardless of the appearance of stained or unstained tissue, the cell bodies of neurons or axons can be located in discrete anatomical structures that need to be named. Those names are specific to whether the structure is central or peripheral. A localized collection of neuron cell bodies in the CNS is referred to as a nucleus. In the PNS, a cluster of neuron cell bodies is referred to as a ganglion. Figure 12.4 indicates how the term nucleus has a few different meanings within anatomy and physiology. It is the center of an atom, where protons and neutrons are found. It is the center of a cell, where the DNA is found. And it is a center of some function in the CNS. There is also a potentially confusing use of the word ganglion, plural equals ganglia, that has a historical explanation. In the central nervous system, there is a group of nuclei that are connected together and were once called the basal ganglia before ganglion became accepted as a description for a peripheral structure. Some sources refer to this group of nuclei as the basal nuclei, to avoid confusion. Figure 12.4 What is a nucleus? A. The nucleus of an atom contains its protons and neutrons. B. The nucleus of a cell is the organelle that contains DNA. C. A nucleus in the CNS is a localized center of function with the cell bodies of several neurons, shown here circled in red. Credit C. Was a B. Wikimedia Commons. Terminology applied to bundles of axons also differs depending on location. A bundle of axons, or fibers, found in the CNS is called a tract, whereas the same thing in the PNS would be called a nerve. There is an important point to make about these terms. which is that they can both be used to refer to the same bundle of axons. When those axons are in the PNS, the term is nerve, but if they are CNS, the term is tract. The most obvious example of this is the axons that project from the retina into the brain. Those axons are called the optic nerve as they leave the eye, but when they are inside the cranium, they are referred to as the optic tract. There is a specific place where the name changes, which is the optic chiasm, but they are still the same axons, figure 12.5. A similar situation outside of science can be described for some roads. Imagine a road called Broad Street in a town. Called Innival. The road leaves Innival and goes to the next town over, called Hometown. When the road crosses the line between the two towns and is in Hometown, its name changes to Main Street. That is the idea behind the naming of the retinal axons. In the PNS, they are called the optic nerve, and in the CNS, they are the optic tract. Table 12.1 helps to clarify which of these terms apply to the central or peripheral nervous systems. Figure 12.5 Optic nerve versus optic tract. This drawing of the connections of the eye to the brain shows the optic nerve extending from the eye to the chiasm, where the structure continues as the optic tract. The same axons extend from the eye to the brain through these two bundles of fibers, but the chiasm represents the border between peripheral and central. In 2003, the Nobel Prize in Physiology or Medicine was awarded to Paul C. Lauterbur and Sir Peter Mansfield for discoveries related to magnetic resonance imaging, MRI. This is a tool to see the structures of the body, not just the nervous system, that depends on magnetic fields associated with certain atomic nuclei. The utility of this technique in the nervous system is that fat tissue and water appear as different shades between black and white. Because white matter is fatty, from myelin, and gray matter is not, they can be easily distinguished in MRI images. Visit the Nobel Prize website, http://openstaxcollege.org/l/nobel_2 to play an interactive game that demonstrates the use of this technology and compares it with other types of imaging technologies. Also, the results from an MRI session are compared with images obtained from X-ray or computed tomography. How do the imaging techniques shown in this game indicate the separation of white and gray matter compared with the freshly dissected tissue shown earlier? Structures of the CNS and PNS group of neuron cell bodies, i.e., gray matter, nucleus ganglion bundle of axons, i.e., white matter, tract nerve. Table 12.1 Functional divisions of the nervous system. The nervous system can also be divided on the basis of its functions, but anatomical divisions and functional divisions are different. The CNS and the PNS both contribute to the same functions, but those functions can be attributed to different regions of the brain, such as the cerebral cortex or the hypothalamus, or to different ganglia in the periphery. The problem with trying to fit functional differences into anatomical divisions is that sometimes the same structure can be part of several functions. For example, the optic nerve carries signals from the retina that are either used for the conscious perception of visual stimuli, which takes place in the cerebral cortex, or for the reflexive responses of smooth muscle tissue that are processed through the hypothalamus. There are two ways to consider how the nervous system is divided functionally. First, the basic functions of the nervous system are sensation, integration, and response. Secondly, control of the body can be somatic or autonomic, divisions that are largely defined by the structures that are involved in the response. There is also a region of the peripheral nervous system that is called the enteric nervous system that is responsible for a specific set of the functions within the realm of autonomic control related to gastrointestinal functions. Basic functions. The nervous system is involved in receiving information about the environment around us, sensation, and generating responses to that information, motor responses. The nervous system can be divided into regions that are responsible for sensation, sensory functions, and for the response, motor functions. But there is a third function that needs to be included. Sensory input needs to be integrated with other sensations, as well as with memories, emotional state, or learning, cognition. Some regions of the nervous system are termed integration or association areas. The process of integration combines sensory perceptions and higher cognitive functions such as memories, learning, and emotion to produce a response. Sensation. The first major function of the nervous system is sensation. Receiving information about the environment to gain input about what is happening outside the body, or, sometimes, within the body. The sensory functions of the nervous system register the presence of a change from homeostasis or a particular event in the environment, known as a stimulus. The senses we think of most are the Big Five: taste, smell, touch, sight, and hearing. The stimuli for taste and smell are both chemical substances, molecules, compounds, ions, etc. Touch is physical or mechanical stimuli that interact with the skin, sight is light stimuli, and hearing is the perception of sound, which is a physical stimulus similar to some aspects of touch. There are actually more senses than just those, but that list represents the major senses. Those five are all senses that receive stimuli from the outside world, and of which there is conscious perception. Additional sensory stimuli might be from the internal environment, inside the body, such as the stretch of an organ wall or the concentration of certain ions in the blood. Response. The nervous system produces a response on the basis of the stimuli perceived by sensory structures. An obvious response would be the movement of muscles, such as withdrawing a hand from a hot stove, but there are broader uses of the term. The nervous system can cause the contraction of all three types of muscle tissue. For example, skeletal muscle contracts to move the skeleton. Cardiac muscle is influenced as heart rate increases during exercise, and smooth muscle contracts as the digestive system moves food along the digestive tract. Responses also include the neural control of glands in the body as well, such as the production and secretion of sweat by the eccrine and merocrine sweat glands found in the skin to lower body temperature. Responses can be divided into those that are voluntary or conscious, contraction of skeletal muscle, and those that are involuntary, contraction of smooth muscles, regulation of cardiac muscle, activation of glands. Voluntary responses are governed by the somatic nervous system and involuntary responses are governed by the autonomic nervous system, which are discussed in the next section. Integration. Stimuli that are received by sensory structures are communicated to the nervous system where that information is processed. This is called integration. Stimuli are compared with, or integrated with, other stimuli, memories of previous stimuli, or the state of a person at a particular time. This leads to the specific response that will be generated. Seeing a baseball pitch to a batter will not automatically cause the batter to swing. The trajectory of the ball and its speed will need to be considered. Maybe the count is 3 balls and 1 strike, and the batter wants to let this pitch go by in the hope of getting a walk to first base. Or maybe the batter's team is so far ahead, it would be fun to just swing away. Controlling the body. The nervous system can be divided into two parts mostly on the basis of a functional difference in responses. The somatic nervous system, SNS, is responsible for conscious perception and voluntary motor responses. Voluntary motor response means the contraction of skeletal muscle, but those contractions are not always voluntary in the sense that you have to want to perform them. Some somatic motor responses are reflexes, and often happen without a conscious decision to perform them. If your friend jumps out from behind a corner and yells, "Boo!" you will be startled and you might scream or leap back. You didn't decide to do that, and you may not have wanted to give your friend a reason to laugh at your expense, but it is a reflex involving skeletal muscle contractions. Other motor responses become automatic, in other words, unconscious, as a person learns motor skills, referred to as habit learning or procedural memory. The autonomic nervous system, ANS, is responsible for involuntary control of the body, usually for the sake of homeostasis, regulation of the internal environment. Sensory input for autonomic functions can be from sensory structures tuned to external or internal environmental stimuli. The motor output extends to smooth and cardiac muscle as well as glandular tissue. The role of the autonomic system is to regulate the organ systems of the body, which usually means to control homeostasis. Sweat glands, for example, are controlled by the autonomic system. When you are hot, sweating helps cool your body down. That is a homeostatic mechanism. But when you are nervous, you might start sweating also. That is not homeostatic. It is the physiological response to an emotional state. There is another division of the nervous system that describes functional responses. The enteric nervous system, ENS, is responsible for controlling the smooth muscle and glandular tissue in your digestive system. It is a large part of the PNS, and is not dependent on the CNS. It is sometimes valid, however, to consider the enteric system to be a part of the autonomic system because the neural structures that make up the enteric system are a component of the autonomic output that regulates digestion. There are some differences between the two, but for our purposes here there will be a good bit of overlap. See Figure 12.6 for examples of where these divisions of the nervous system can be found. Figure 12.6 Somatic, autonomic, and enteric structures of the nervous system. Somatic structures include the spinal nerves, both motor and sensory fibers, as well as the sensory ganglia, posterior root ganglia and cranial nerve ganglia. Autonomic structures are found in the nerves also, but include the sympathetic and parasympathetic ganglia. The enteric nervous system includes the nervous tissue within the organs of the digestive tract. Visit this site, http://openstaxcollege.org/l/troublestairs, to read about a woman that notices that her daughter is having trouble walking up the stairs. This leads to the discovery of a hereditary condition that affects the brain and spinal cord. The electromyography and MRI tests indicated deficiencies in the spinal cord and cerebellum, both of which are responsible for controlling coordinated movements. To what functional division of the nervous system would these structures belong? How much of your brain do you use? Have you ever heard the claim that humans only use 10% of their brains? Maybe you have seen an advertisement on a website saying that there is a secret to unlocking the full potential of your mind, as if there were 90% of your brain sitting idle, just waiting for you to use it. If you see an ad like that, don't click. It isn't true. An easy way to see how much of the brain a person uses is to take measurements of brain activity while performing a task. An example of this kind of measurement is functional magnetic resonance imaging, fMRI, which generates a map of the most active areas and can be generated and presented in 3 dimensions. Figure 12.7. This procedure is different from the standard MRI technique because it is measuring changes in the tissue in time with an experimental condition or event. Figure 12.7 fMRI. This fMRI shows activation of the visual cortex in response to visual stimuli. Credit. Superborsic, Wikimedia Commons. The underlying assumption is that active nervous tissue will have greater blood flow. By having the subject perform a visual task, activity all over the brain can be measured. Consider this possible experiment. The subject is told to look at a screen with a black dot in the middle, a fixation point. A photograph of a face is projected on the screen away from the center. The subject has to look at the photograph and decipher what it is. The subject has been instructed to push a button if the photograph is of someone they recognize. The photograph might be of a celebrity, so the subject would press the button, or it might be of a random person unknown to the subject, so the subject would not press the button. In this task, visual sensory areas would be active, integrating areas would be active, motor areas responsible for moving the eyes would be active, and motor areas for pressing the button with a finger would be active. Those areas are distributed all around the brain, and the fMRI images would show activity in more than just 10% of the brain. Some evidence suggests that about 80% of the brain is using energy, based on blood flow to the tissue, during well-defined tasks similar to the one suggested above. This task does not even include all of the functions the brain performs. There is no language response. The body is mostly lying still in the MRI machine, and it does not consider the autonomic functions that would be ongoing in the background. 12.2. Nervous tissue. By the end of this section, you will be able to describe the basic structure of a neuron. Identify the different types of neurons on the basis of polarity. List the glial cells of the CNS and describe their function. List the glial cells of the PNS and describe their function. Nervous tissue is composed of two types of cells, neurons and glial cells. Neurons are the primary type of cell that most anyone associates with the nervous system. They are responsible for the computation and communication that the nervous system provides. They are electrically active and release chemical signals to target cells. Glial cells, or glia, are known to play a supporting role for nervous tissue. Ongoing research pursues an expanded role that glial cells might play in signaling, but neurons are still considered the basis of this function. Neurons are important, but without glial support they would not be able to perform their function. Neurons are the cells considered to be the basis of nervous tissue. They are responsible for the electrical signals that communicate information about sensations, and that produce movements in response to those stimuli, along with inducing thought processes within the brain. An important part of the function of neurons is in their structure, or shape. The three-dimensional shape of these cells makes the immense numbers of connections within the nervous system possible. Parts of a neuron. As you learned in the first section, the main part of a neuron is the cell body, which is also known as the soma. Soma equals body. The cell body contains the nucleus and most of the major organelles. But what makes neurons special is that they have many extensions of their cell membranes, which are generally referred to as processes. Neurons are usually described as having one, and only one, axon, a fiber that emerges from the cell body and projects to target cells. That single axon can branch repeatedly to communicate with many target cells. It is the axon that propagates the nerve impulse, which is communicated to one or more cells. The other processes of the neuron are dendrites, which receive information from other neurons at specialized areas of contact called synapses. The dendrites are usually highly branched processes, providing locations for other neurons to communicate with the cell body. Information flows through a neuron from the dendrites, across the cell body, and down the axon. This gives the neuron a polarity, meaning that information flows in this one direction. Figure 12.8 shows the relationship of these parts to one another. Figure 12.8 Parts of a neuron. The major parts of the neuron are labeled on a multipolar neuron from the CNS. Where the axon emerges from the cell body, there is a special region referred to as the axon hillock. This is a tapering of the cell body toward the axon fiber. Within the axon hillock, the cytoplasm changes to a solution of limited components called axoplasm. Because the axon hillock represents the beginning of the axon, it is also referred to as the initial segment. Many axons are wrapped by an insulating substance called myelin, which is actually made from glial cells. Myelin acts as insulation much like the plastic or rubber that is used to insulate electrical wires. A key difference between myelin and the insulation on a wire is that there are gaps in the myelin covering of an axon. Each gap is called a node of Ranvier and is important to the way that electrical signals travel down the axon. The length of the axon between each gap, which is wrapped in myelin, is referred to as an axon segment. At the end of the axon is the axon terminal, where there are usually several branches extending toward the target cell, each of which ends in an enlargement called a synaptic end bulb. These bulbs are what make the connection with the target cell at the synapse. Visit this site, http://openstaxcollege.org/l/nervetissue3 to learn about how nervous tissue is composed of neurons and glial cells. Neurons are dynamic cells with the ability to make a vast number of connections, to respond incredibly quickly to stimuli, and to initiate movements on the basis of those stimuli. They are the focus of intense research because failures in physiology can lead to devastating illnesses. Why are neurons only found in animals? Based on what this article says about neuron function, why wouldn't they be helpful for plants or microorganisms? Types of neurons. There are many neurons in the nervous system, a number in the trillions. And there are many different types of neurons. They can be classified by many different criteria. The first way to classify them is by the number of processes attached to the cell body. Using the standard model of neurons, one of these processes is the axon, and the rest are dendrites. Because information flows through the neuron from dendrites or cell bodies toward the axon, these names are based on the neuron's polarity. Figure 12.9. Figure 12.9 Neuron classification by shape. Unipolar cells have one process that includes both the axon and dendrite. Bipolar cells have two processes, the axon and a dendrite. Multipolar cells have more than two processes, the axon and two or more dendrites. Unipolar cells have only one process emerging from the cell. True unipolar cells are only found in invertebrate animals, so the unipolar cells in humans are more appropriately called pseudo-unipolar cells. Invertebrate unipolar cells do not have dendrites. Human unipolar cells have an axon that emerges from the cell body, but it splits so that the axon can extend along a very long distance. At one end of the axon are dendrites, and at the other end, the axon forms synaptic connections with a target. Unipolar cells are exclusively sensory neurons and have two unique characteristics. First, their dendrites are receiving sensory information, sometimes directly from the stimulus itself. Secondly, the cell bodies of unipolar neurons are always found in ganglia. Sensory reception is a peripheral function. Those dendrites are in the periphery, perhaps in the skin, so the cell body is in the periphery, though closer to the CNS in a ganglion. The axon projects from the dendrite endings, past the cell body in a ganglion, and into the central nervous system. Bipolar cells have two processes, which extend from each end of the cell body, opposite to each other. One is the axon and one the dendrite. Bipolar cells are not very common. They are found mainly in the olfactory epithelium, where smell stimuli are sensed, and as part of the retina. Multipolar neurons are all of the neurons that are not unipolar or bipolar. They have one axon and two or more dendrites, usually many more. With the exception of the unipolar sensory ganglion cells, and the two specific bipolar cells mentioned above, all other neurons are multipolar. Some cutting-edge research suggests that certain neurons in the CNS do not conform to the standard model of one, and only one, axon. Some sources describe a fourth type of neuron, called an anaxonic neuron. The name suggests that it has no axon, and equals, without, but this is not accurate. Anaxonic neurons are very small, and if you look through a microscope at the standard resolution used in histology, approximately 400x to 1000x total magnification, you will not be able to distinguish any process specifically as an axon or a dendrite. Any of those processes can function as an axon depending on the conditions at any given time. Nevertheless, even if they cannot be easily seen, and one specific process is definitively the axon, these neurons have multiple processes and are therefore multipolar. Neurons can also be classified on the basis of where they are found, who found them, what they do, or even what chemicals they use to communicate with each other. Some neurons referred to in this section on the nervous system are named on the basis of those sorts of classifications. Figure 12.10. For example, a multipolar neuron that has a very important role to play in a part of the brain called the cerebellum is known as a Purkinje, commonly pronounced Purkinje, cell. It is named after the anatomist who discovered it, Jan Evangelista Purkinje, 1787 to 1869. Figure 12.10 Other neuron classifications. 3 examples of neurons that are classified on the basis of other criteria. A. The pyramidal cell is a multipolar cell with a cell body that is shaped something like a pyramid. B. The Purkinje cell in the cerebellum was named after the scientist who originally described it. C. Olfactory neurons are named for the functional group with which they belong. Glial cells, or neuroglia or simply glia, are the other type of cell found in nervous tissue. They are considered to be supporting cells, and many functions are directed at helping neurons complete their function for communication. The name glia comes from the Greek word that means glue, and was coined by the German pathologist Rudolf Virchow, who wrote in 1856, This connective substance, which is in the brain, the spinal cord, and the special sense nerves, is a kind of glue, neuroglia, in which the nervous elements are planted. Today, research into nervous tissue has shown that there are many deeper roles that these cells play. And research may find much more about them in the future. There are 6 types of glial cells. 4 of them are found in the CNS and 2 are found in the PNS. Table 12.2 outlines some common characteristics and functions. Glial cell types by location and basic function: CNS glia, PNS glia. Basic function: astrocyte satellite cell support, oligodendrocyte Schwann cell insulation, myelination, microglia, immune surveillance and phagocytosis, ependymal cell, creating CSF. Table 12.2 glial cells of the CNS. One cell providing support to neurons of the CNS is the astrocyte, so named because it appears to be star-shaped under the microscope. Astro equals star. Astrocytes have many processes extending from their main cell body, not axons or dendrites like neurons, just cell extensions. Those processes extend to interact with neurons, blood vessels, or the connective tissue covering the CNS that is called the pia mater. Figure 12.11. Generally, they are supporting cells for the neurons in the central nervous system. Some ways in which they support neurons in the central nervous system are by maintaining the concentration of chemicals in the extracellular space, removing excess signaling molecules, reacting to tissue damage, and contributing to the blood-brain barrier, BBB. The blood-brain barrier is a physiological barrier that keeps many substances that circulate in the rest of the body from getting into the central nervous system, restricting what can cross from circulating blood into the CNS. Nutrient molecules, such as glucose or amino acids, can pass through the BBB, but other molecules cannot. This actually causes problems with drug delivery to the CNS. Pharmaceutical companies are challenged to design drugs that can cross the BBB as well as have an effect on the nervous system. Figure 12.11 glial cells of the CNS. The CNS has astrocytes, oligodendrocytes, microglia, and ependymal cells that support the neurons of the CNS in several ways. Like a few other parts of the body, the brain has a privileged blood supply. Very little can pass through by diffusion. Most substances that cross the wall of a blood vessel into the CNS must do so through an active transport process. Because of this, only specific types of molecules can enter the CNS. Glucose, the primary energy source, is allowed, as are amino acids. Water and some other small particles, like gases and ions, can enter. But most everything else cannot, including white blood cells, which are one of the body's main lines of defense. While this barrier protects the CNS from exposure to toxic or pathogenic substances, it also keeps out the cells that could protect the brain and spinal cord from disease and damage. The BBB also makes it harder for pharmaceuticals to be developed that can affect the nervous system. Aside from finding efficacious substances, the means of delivery is also crucial. Also found in CNS tissue is the oligodendrocyte, sometimes called just oligo, which is the glial cell type that insulates axons in the CNS. The name means cell of a few branches. Oligo equals few. Dendro equals branches, cyte equals cell. There are a few processes that extend from the cell body. Each one reaches out and surrounds an axon to insulate it in myelin. One oligodendrocyte will provide the myelin for multiple axon segments, either for the same axon or for separate axons. The function of myelin will be discussed below. Microglia are, as the name implies, smaller than most of the other glial cells. Ongoing research into these cells, although not entirely conclusive, suggests that they may originate as white blood cells, called macrophages, that become part of the CNS during early development. While their origin is not conclusively determined, their function is related to what macrophages do in the rest of the body. When macrophages encounter diseased or damaged cells in the rest of the body, they ingest and digest those cells or the pathogens that cause disease. Microglia are the cells in the CNS that can do this in normal, healthy tissue, and they are therefore also referred to as CNS-resident macrophages. The ependymal cell is a glial cell that filters blood to make cerebrospinal fluid, CSF, the fluid that circulates through the CNS. Because of the privileged blood supply inherent in the BBB, the extracellular space in nervous tissue does not easily exchange components with the blood. Ependymal cells line each ventricle, one of 4 central cavities that are remnants of the hollow center of the neural tube formed during the embryonic development of the brain. The choroid plexus is a specialized structure in the ventricles where ependymal cells come in contact with blood vessels and filter and absorb components of the blood to produce cerebrospinal fluid. Because of this, ependymal cells can be considered a component of the BBB, or a place where the BBB breaks down. These glial cells appear similar to epithelial cells, making a single layer of cells with little intracellular space and tight connections between adjacent cells. They also have cilia on their apical surface to help move the CSF through the ventricular space. The relationship of these glial cells to the structure of the CNS is seen in Figure 12.11. Glial cells of the PNS. One of the 2 types of glial cells found in the PNS is the satellite cell. Satellite cells are found in sensory and autonomic ganglia, where they surround the cell bodies of neurons. This accounts for the name, based on their appearance under the microscope. They provide support, performing similar functions in the periphery as astrocytes do in the CNS, except, of course, for establishing the BBB. The second type of glial cell is the Schwann cell, which insulate axons with myelin in the periphery. Schwann cells are different than oligodendrocytes, in that a Schwann cell wraps around a portion of only one axon segment and no others. Oligodendrocytes have processes that reach out to multiple axon segments, whereas the entire Schwann cell surrounds just one axon segment. The nucleus and cytoplasm of the Schwann cell are on the edge of the myelin sheath. The relationship of these 2 types of glial cells to ganglia and nerves in the PNS is seen in Figure 12.12. Figure 12.12 Glial cells of the PNS. The PNS has satellite cells and Schwann cells. Myelin. The insulation for axons in the nervous system is provided by glial cells, oligodendrocytes in the CNS, and Schwann cells in the PNS. Whereas the manner in which either cell is associated with the axon segment, or segments, that it insulates is different, the means of myelinating an axon segment is mostly the same in the 2 situations. Myelin is a lipid-rich sheath that surrounds the axon and by doing so creates a myelin sheath that facilitates the transmission of electrical signals along the axon. The lipids are essentially the phospholipids of the glial cell membrane. Myelin, however, is more than just the membrane of the glial cell. It also includes important proteins that are integral to that membrane. Some of the proteins help to hold the layers of the glial cell membrane closely together. The appearance of the myelin sheath can be thought of as similar to the pastry wrapped around a hot dog for pigs in a blanket, or a similar food. The glial cell is wrapped around the axon several times with little to no cytoplasm between the glial cell layers. For oligodendrocytes, the rest of the cell is separate from the myelin sheath as a cell process extends back toward the cell body. A few other processes provide the same insulation for other axon segments in the area. For Schwann cells, the outermost layer of the cell membrane contains cytoplasm and the nucleus of the cell is a bulge on one side of the myelin sheath. During development, the glial cell is loosely or incompletely wrapped around the axon, Figure 12.13a. The edges of this loose enclosure extend toward each other, and one end tucks under the other. The inner edge wraps around the axon, creating several layers, and the other edge closes around the outside so that the axon is completely enclosed. View the University of Michigan web scope, http://openstaxcollege.org/l/nerve fiber to see an electron micrograph of a cross-section of a myelinated nerve fiber. The axon contains microtubules and neurofilaments that are bounded by a plasma membrane known as the axonema. Outside the plasma membrane of the axon is the myelin sheath, which is composed of the tightly wrapped plasma membrane of a Schwann cell. What aspects of the cells in this image react with the stain to make them a deep, dark, black color, such as the multiple layers that are the myelin sheath? Myelin sheaths can extend for 1 or 2 millimeters, depending on the diameter of the axon. Axon diameters can be as small as 1 to 20 micrometers. Because a micrometer is 1/1000th of a millimeter, this means that the length of a myelin sheath can be 100,000 times the diameter of the axon. Figure 12.8, Figure 12.11, and Figure 12.12 show the myelin sheath surrounding an axon segment, but are not to scale. If the myelin sheath were drawn to scale, the neuron would have to be immense, possibly covering an entire wall of the room in which you are sitting. Figure 12.13 The process of myelination myelinating glia wraps several layers of cell membrane around the cell membrane of an axon segment. A single Schwann cell insulates a segment of a peripheral nerve, whereas in the CNS, an oligodendrocyte may provide insulation for a few separate axon segments. M × 1,460,000. Micrograph provided by the Regents of University of Michigan Medical School. Copyright 2012. Nervous tissue. Several diseases can result from the demyelination of axons. The causes of these diseases are not the same. Some have genetic causes, some are caused by pathogens, and others are the result of autoimmune disorders. Though the causes are varied, the results are largely similar. The myelin insulation of axons is compromised, making electrical signaling slower. Multiple sclerosis, MS, is one such disease. It is an example of an autoimmune disease. The antibodies produced by lymphocytes, a type of white blood cell, mark myelin as something that should not be in the body. This causes inflammation and the destruction of the myelin in the central nervous system. As the insulation around the axons is destroyed by the disease, scarring becomes obvious. This is where the name of the disease comes from. Sclerosis means hardening of tissue, which is what a scar is. Multiple scars are found in the white matter of the brain and spinal cord. The symptoms of MS include both somatic and autonomic deficits. Control of the musculature is compromised, as is control of organs such as the bladder. Guillain-Barré, pronounced Guillain-Barré, syndrome is an example of a demyelinating disease of the peripheral nervous system. It is also the result of an autoimmune reaction, but the inflammation is in peripheral nerves. Sensory symptoms or motor deficits are common, and autonomic failures can lead to changes in the heart rhythm or a drop in blood pressure, especially when standing, which causes dizziness. 12.3. The function of nervous tissue. By the end of this section, you will be able to distinguish the major functions of the nervous system: sensation, integration, and response. List the sequence of events in a simple sensory receptor motor response pathway. Having looked at the components of nervous tissue and the basic anatomy of the nervous system, next comes an understanding of how nervous tissue is capable of communicating within the nervous system. Before getting to the nuts and bolts of how this works, an illustration of how the components come together will be helpful. An example is summarized in Figure 12.14. Figure 12.14 Testing the water. 1. The sensory neuron has endings in the skin that sense a stimulus such as water temperature. The strength of the signal that starts here is dependent on the strength of the stimulus. 2. The graded potential from the sensory endings, if strong enough, will initiate an action potential at the initial segment of the axon, which is immediately adjacent to the sensory endings in the skin. 3. The axon of the peripheral sensory neuron enters the spinal cord and contacts another neuron in the gray matter. The contact is a synapse where another graded potential is caused by the release of a chemical signal from the axon terminals. 4. An action potential is initiated at the initial segment of this neuron and travels up the sensory pathway to a region of the brain called the thalamus. Another synapse passes the information along to the next neuron. 5. The sensory pathway ends when the signal reaches the cerebral cortex. 6. After integration with neurons in other parts of the cerebral cortex, a motor command is sent from the precentral gyrus of the frontal cortex. 7. The upper motor neuron sends an action potential down to the spinal cord. The target of the upper motor neuron is the dendrites of the lower motor neuron in the gray matter of the spinal cord. 8. The axon of the lower motor neuron emerges from the spinal cord in a nerve and connects to a muscle through a neuromuscular junction to cause contraction of the target muscle. Imagine you are about to take a shower in the morning before going to school. You have turned on the faucet to start the water as you prepare to get in the shower. After a few minutes, you expect the water to be a temperature that will be comfortable to enter. So you put your hand out into the spray of water. What happens next depends on how your nervous system interacts with the stimulus of the water temperature and what you do in response to that stimulus. Found in the skin of your fingers or toes is a type of sensory receptor that is sensitive to temperature, called a thermoreceptor. When you place your hand under the shower, figure 12.15, the cell membrane of the thermoreceptors changes its electrical state, voltage. The amount of change is dependent on the strength of the stimulus, how hot the water is. This is called a graded potential. If the stimulus is strong, the voltage of the cell membrane will change enough to generate an electrical signal that will travel down the axon. You have learned about this type of signaling before, with respect to the interaction of nerves and muscles at the neuromuscular junction. The voltage at which such a signal is generated is called the threshold, and the resulting electrical signal is called an action potential. In this example, the action potential travels, a process known as propagation, along the axon from the axon hillock to the axon terminals and into the synaptic end bulbs. When this signal reaches the end bulbs, it causes the release of a signaling molecule called a neurotransmitter. Figure 12.15. The sensory input receptors in the skin sense the temperature of the water. The neurotransmitter diffuses across the short distance of the synapse and binds to a receptor protein of the target neuron. When the molecular signal binds to the receptor, the cell membrane of the target neuron changes its electrical state and a new graded potential begins. If that graded potential is strong enough to reach threshold, the second neuron generates an action potential at its axon hillock. The target of this neuron is another neuron in the thalamus of the brain, the part of the CNS that acts as a relay for sensory information. At another synapse, neurotransmitter is released and binds to its receptor. The thalamus then sends the sensory information to the cerebral cortex, the outermost layer of gray matter in the brain, where conscious perception of that water temperature begins. Within the cerebral cortex, information is processed among many neurons, integrating the stimulus of the water temperature with other sensory stimuli. With your emotional state, you just aren't ready to wake up. The bed is calling to you, memories, perhaps of the lab notes you have to study before a quiz. Finally, a plan is developed about what to do, whether that is to turn the temperature up, turn the whole shower off and go back to bed, or step into the shower. To do any of these things, the cerebral cortex has to send a command out to your body to move muscles. Figure 12.16. Figure 12.16. The motor response on the basis of the sensory input and the integration in the CNS. A motor response is formulated and executed. A region of the cortex is specialized for sending signals down to the spinal cord for movement. The upper motor neuron is in this region, called the precentral gyrus of the frontal cortex, which has an axon that extends all the way down the spinal cord. At the level of the spinal cord at which this axon makes a synapse, a graded potential occurs in the cell membrane of a lower motor neuron. This second motor neuron is responsible for causing muscle fibers to contract. In the manner described in the chapter on muscle tissue, an action potential travels along the motor neuron axon into the periphery. The axon terminates on muscle fibers at the neuromuscular junction. Acetylcholine is released at this specialized synapse, which causes the muscle action potential to begin, following a large potential known as an end plate potential. When the lower motor neuron excites the muscle fiber, it contracts. All of this occurs in a fraction of a second, but this story is the basis of how the nervous system functions. Neurophysiologist understanding how the nervous system works could be a driving force in your career. Studying neurophysiology is a very rewarding path to follow. It means that there is a lot of work to do, but the rewards are worth the effort. The career path of a research scientist can be straightforward: college, graduate school, postdoctoral research, academic research position at a university. A bachelor's degree in science will get you started, and for neurophysiology that might be in biology, psychology, computer science, engineering, or neuroscience. But the real specialization comes in graduate school. There are many different programs out there to study the nervous system, not just neuroscience itself. Most graduate programs are doctoral, meaning that a master's degree is not part of the work. These are usually considered 5-year programs, with the first 2 years dedicated to coursework and finding a research mentor, and the last 3 years dedicated to finding a research topic and pursuing that with a near single-mindedness. The research will usually result in a few publications in scientific journals, which will make up the bulk of a doctoral dissertation. After graduating with a PhD, researchers will go on to find specialized work called a postdoctoral fellowship within established labs. In this position, a researcher starts to establish their own research career with the hopes of finding an academic position at a research university. Other options are available if you are interested in how the nervous system works. Especially for neurophysiology, a medical degree might be more suitable so you can learn about the clinical applications of neurophysiology and possibly work with human subjects. An academic career is not a necessity. Biotechnology firms are eager to find motivated scientists ready to tackle the tough questions about how the nervous system works so that therapeutic chemicals can be tested on some of the most challenging disorders such as Alzheimer's disease or Parkinson's disease, or spinal cord injury. Others with a medical degree and a specialization in neuroscience go on to work directly with patients, diagnosing and treating mental disorders. You can do this as a psychiatrist, a neuropsychologist, a neuroscience nurse, or a neurodiagnostic technician, among other possible career paths. 12.4. The action potential. By the end of this section, you will be able to describe the components of the membrane that establish the resting membrane potential. Describe the changes that occur to the membrane that result in the action potential. The functions of the nervous system, sensation, integration, and response, depend on the functions of the neurons underlying these pathways. To understand how neurons are able to communicate, it is necessary to describe the role of an excitable membrane in generating these signals. The basis of this communication is the action potential, which demonstrates how changes in the membrane can constitute a signal. Looking at the way these signals work in more variable circumstances involves a look at graded potentials, which will be covered in the next section. Electrically active cell membranes. Most cells in the body make use of charged particles, ions, to build up a charge across the cell membrane. Previously, this was shown to be a part of how muscle cells work. For skeletal muscles to contract, based on excitation-contraction coupling, requires input from a neuron. Both of the cells make use of the cell membrane to regulate ion movement between the extracellular fluid and cytosol. As you learned in the chapter on cells, the cell membrane is primarily responsible for regulating what can cross the membrane and what stays on only one side. The cell membrane is a phospholipid bilayer, so only substances that can pass directly through the hydrophobic core can diffuse through unaided. Charged particles, which are hydrophilic by definition, cannot pass through the cell membrane without assistance. Figure 12.17. Transmembrane proteins, specifically channel proteins, make this possible. Several passive transport channels, as well as active transport pumps, are necessary to generate a transmembrane potential and an action potential. Of special interest is the carrier protein referred to as the sodium-potassium pump that moves sodium ions, Na+, out of a cell and potassium ions, K+, into a cell, thus regulating ion concentration on both sides of the cell membrane. Figure 12.17. Cell membrane and transmembrane proteins. The cell membrane is composed of a phospholipid bilayer and has many transmembrane proteins, including different types of channel proteins that serve as ion channels. The sodium-potassium pump requires energy in the form of adenosine triphosphate, ATP, so it is also referred to as an ATPase. As was explained in the cell chapter, the concentration of Na+ is higher outside the cell than inside, and the concentration of K+ is higher inside the cell than outside. That means that this pump is moving the ions against the concentration gradients for sodium and potassium, which is why it requires energy. In fact, the pump basically maintains those concentration gradients. Ion channels are pores that allow specific charged particles to cross the membrane in response to an existing concentration gradient. Proteins are capable of spanning the cell membrane, including its hydrophobic core, and can interact with the charge of ions because of the varied properties of amino acids found within specific domains or regions of the protein channel. Hydrophobic amino acids are found in the domains that are opposed to the hydrocarbon tails of the phospholipids. Hydrophilic amino acids are exposed to the fluid environments of the extracellular fluid and cytosol. Additionally, the ions will interact with the hydrophilic amino acids, which will be selective for the charge of the ion. Channels for cations, positive ions, will have negatively charged side chains in the pore. Channels for anions, negative ions, will have positively charged side chains in the pore. This is called electrochemical exclusion, meaning that the channel pore is charge-specific. Ion channels can also be specified by the diameter of the pore. The distance between the amino acids will be specific for the diameter of the ion when it dissociates from the water molecules surrounding it. Because of the surrounding water molecules, larger pores are not ideal for smaller ions because the water molecules will interact, by hydrogen bonds, more readily than the amino acid side chains. This is called size exclusion. Some ion channels are selective for charge but not necessarily for size, and thus are called a nonspecific channel. These nonspecific channels allow cations, particularly Na+, K+, and Ca2+, to cross the membrane, but exclude anions. Ion channels do not always freely allow ions to diffuse across the membrane. Some are opened by certain events, meaning the channels are gated. So another way that channels can be categorized is on the basis of how they are gated. Although these classes of ion channels are found primarily in the cells of nervous or muscular tissue, they also can be found in the cells of epithelial and connective tissues. A ligand-gated channel opens because a signaling molecule, a ligand, binds to the extracellular region of the channel. This type of channel is also known as an ionotropic receptor, because when the ligand, known as a neurotransmitter in the nervous system, binds to the protein, ions cross the membrane changing its charge. Figure 12.18. Figure 12.18 ligand-gated channels when the ligand, in this case the neurotransmitter acetylcholine, binds to a specific location on the extracellular surface of the channel protein. The pore opens to allow select ions through. The ions, in this case, are cations of sodium, calcium, and potassium. A mechanically gated channel opens because of a physical distortion of the cell membrane. Many channels associated with the sense of touch, somatosensation, are mechanically gated. For example, as pressure is applied to the skin, these channels open and allow ions to enter the cell. Similar to this type of channel would be the channel that opens on the basis of temperature changes, as in testing the water in the shower. Figure 12.19. Figure 12.19 mechanically gated channels. When a mechanical change occurs in the surrounding tissue, such as pressure or touch, the channel is physically opened. Thermoreceptors work on a similar principle. When the local tissue temperature changes, the protein reacts by physically opening the channel. A voltage-gated channel is a channel that responds to changes in the electrical properties of the membrane in which it is embedded. Normally, the inner portion of the membrane is at a negative voltage. When that voltage becomes less negative, the channel begins to allow ions to cross the membrane. Figure 12.20. Figure 12.20 voltage-gated channels. Voltage-gated channels open when the transmembrane voltage changes around them. Amino acids in the structure of the protein are sensitive to charge and cause the pore to open to the selected ion. A leakage channel is randomly gated, meaning that it opens and closes at random, hence the reference to leaking. There is no actual event that opens the channel. Instead, It has an intrinsic rate of switching between the open and closed states. Leakage channels contribute to the resting transmembrane voltage of the excitable membrane. Figure 12.21. Figure 12.21 leakage channels. In certain situations, ions need to move across the membrane randomly. The particular electrical properties of certain cells are modified by the presence of this type of channel. The membrane potential. The electrical state of the cell membrane can have several variations. These are all variations in the membrane potential. A potential is a distribution of charge across the cell membrane, measured in millivolts, MV. The standard is to compare the inside of the cell relative to the outside, so the membrane potential is a value representing the charge on the intracellular side of the membrane based on the outside being zero. Relatively speaking, figure 12.22. Figure 12.22 measuring charge across a membrane with a voltmeter. A recording electrode is inserted into the cell and a reference electrode is outside the cell. By comparing the charge measured by these two electrodes, the transmembrane voltage is determined. It is conventional to express that value for the cytosol relative to the outside. The concentration of ions in extracellular and intracellular fluids is largely balanced, with a net neutral charge. However, a slight difference in charge occurs right at the membrane surface, both internally and externally. It is the difference in this very limited region that has all the power in neurons and muscle cells to generate electrical signals, including action potentials. Before these electrical signals can be described, the resting state of the membrane must be explained. When the cell is at rest, and the ion channels are closed, except for leakage channels which randomly open, ions are distributed across the membrane in a very predictable way. The concentration of Na+ outside the cell is 10 times greater than the concentration inside. Also, the concentration of K+ inside the cell is greater than outside. The cytosol contains a high concentration of anions, in the form of phosphate ions and negatively charged proteins. Large anions are a component of the inner cell membrane, including specialized phospholipids and proteins associated with the inner leaflet of the membrane. Leaflet is a term used for one side of the lipid bilayer membrane. The negative charge is localized in the large anions. With the ions distributed across the membrane at these concentrations, the difference in charge is measured at -70 mV, the value described as the resting membrane potential. The exact value measured for the resting membrane potential varies between cells, but -70 mV is most commonly used as this value. This voltage would actually be much lower except for the contributions of some important proteins in the membrane. Leakage channels allow Na+ to slowly move into the cell or K+ to slowly move out, and the Na+, K+ pump restores them. This may appear to be a waste of energy, but each has a role in maintaining the membrane potential. The action potential. Resting membrane potential describes the steady state of the cell, which is a dynamic process that is balanced by ion leakage and ion pumping. Without any outside influence, it will not change. To get an electrical signal started, the membrane potential has to change. This starts with a channel opening for Na+ in the membrane. Because the concentration of Na+ is higher outside the cell than inside the cell by a factor of 10, ions will rush into the cell that are driven largely by the concentration gradient. Because sodium is a positively charged ion, it will change the relative voltage immediately inside the cell relative to immediately outside. The resting potential is the state of the membrane at a voltage of -70 mV, so the sodium cation entering the cell will cause it to become less negative. This is known as depolarization, meaning the membrane potential moves toward zero. The concentration gradient for Na+ is so strong that it will continue to enter the cell even after the membrane potential has become zero, so that the voltage immediately around the pore begins to become positive. The electrical gradient also plays a role, as negative proteins below the membrane attract the sodium ion. The membrane potential will reach +30 mV by the time sodium has entered the cell. As the membrane potential reaches +30 mV, other voltage-gated channels are opening in the membrane. These channels are specific for the potassium ion. A concentration gradient acts on K+, as well. As K+ starts to leave the cell, taking a positive charge with it, the membrane potential begins to move back toward its resting voltage. This is called repolarization, meaning that the membrane voltage moves back toward the -70 mV value of the resting membrane potential. Repolarization returns the membrane potential to the -70 mV value that indicates the resting potential, but it actually overshoots that value. Potassium ions reach equilibrium when the membrane voltage is below -70 mV, so a period of hyperpolarization occurs while the K+ channels are open. Those K+ channels are slightly delayed in closing, accounting for this short overshoot. What has been described here is the action potential, which is presented as a graph of voltage over time in Figure 12.23. It is the electrical signal that nervous tissue generates for communication. The change in the membrane voltage from -70 mV at rest to +30 mV at the end of depolarization is a 100 MV change. That can also be written as a 0.1-V change. To put that value in perspective, think about a battery. An AA battery that you might find in a television remote has a voltage of 1.5 volts, or a 9V battery. The rectangular battery with two posts on one end is, obviously, 9V. The change seen in the action potential is 1 or 2 orders of magnitude less than the charge in these batteries. In fact, the membrane potential can be described as a battery. A charge is stored across the membrane that can be released under the correct conditions. A battery in your remote has stored a charge that is released when you push a button. Figure 12.23 graph of action potential plotting voltage measured across the cell membrane against time. The action potential begins with depolarization, followed by repolarization, which goes past the resting potential into hyperpolarization, and finally the membrane returns to rest. What happens across the membrane of an electrically active cell is a dynamic process that is hard to visualize with static images or through text descriptions. View this animation. http://openstaxcollege.org/l/dynamic1 to learn more about this process. What is the difference between the driving force for Na+ and K+? And what is similar about the movement of these two ions? The question is, now, what initiates the action potential? The description above conveniently glosses over that point. But it is vital to understanding what is happening. The membrane potential will stay at the resting voltage until something changes. The description above just says that a Na+ channel opens. Now, to say, a channel opens, does not mean that one individual transmembrane protein changes. Instead, it means that one kind of channel opens. There are a few different types of channels that allow Na+ to cross the membrane. A ligand-gated Na+ channel will open when a neurotransmitter binds to it, and a mechanically gated Na+ channel will open when a physical stimulus affects a sensory receptor, like pressure applied to the skin compresses a touch receptor. Whether it is a neurotransmitter binding to its receptor protein or a sensory stimulus activating a sensory receptor cell, some stimulus gets the process started. Sodium starts to enter the cell and the membrane becomes less negative. A third type of channel that is an important part of depolarization in the action potential is the voltage-gated Na+ channel. The channels that start depolarizing the membrane because of a stimulus help the cell to depolarize from -70 mV to -55 mV. Once the membrane reaches that voltage, the voltage-gated Na+ channels open. This is what is known as the threshold. Any depolarization that does not change the membrane potential to -55 mV or higher will not reach threshold and thus will not result in an action potential. Also, any stimulus that depolarizes the membrane to -55 mV or beyond will cause a large number of channels to open and an action potential will be initiated. Because of the threshold, the action potential can be likened to a digital event. It either happens or it does not. If the threshold is not reached, then no action potential occurs. If depolarization reaches -55 mV, then the action potential continues and runs all the way to plus 30 millivolts, at which K+ causes repolarization, including the hyperpolarizing overshoot. Also, those changes are the same for every action potential, which means that once the threshold is reached, the exact same thing happens. A stronger stimulus, which might depolarize the membrane well past threshold, will not make a bigger action potential. Action potentials are all or none. Either the membrane reaches the threshold and everything occurs as described above, or the membrane does not reach the threshold and nothing else happens. All action potentials peak at the same voltage, plus 30 millivolts, so one action potential is not bigger than another. Stronger stimuli will initiate multiple action potentials more quickly, but the individual signals are not bigger. Thus, for example, you will not feel a greater sensation of pain, or have a stronger muscle contraction, because of the size of the action potential because they are not different sizes. As we have seen, the depolarization and repolarization of an action potential are dependent on two types of channels: the voltage-gated Na+ channel and the voltage-gated K+ channel. The voltage-gated Na+ channel actually has two gates. One is the activation gate, which opens when the membrane potential crosses -55 millivolts. The other gate is the inactivation gate, which closes after a specific period of time, on the order of a fraction of a millisecond. When a cell is at rest, the activation gate is closed and the inactivation gate is open. However, when the threshold is reached, the activation gate opens, allowing Na+ to rush into the cell. Timed with the peak of depolarization, the inactivation gate closes. During repolarization, no more sodium can enter the cell. When the membrane potential passes -55 millivolts again, the activation gate closes. After that, the inactivation gate reopens, making the channel ready to start the whole process over again. The voltage-gated K+ channel has only one gate, which is sensitive to a membrane voltage of -50 millivolts. However, it does not open as quickly as the voltage-gated Na+ channel does. It might take a fraction of a millisecond for the channel to open once that voltage has been reached. The timing of this coincides exactly with when the Na+ flow peaks, so voltage-gated K+ channels open just as the voltage-gated Na+ channels are being inactivated. As the membrane potential repolarizes and the voltage passes -50 millivolts again, the channel closes, again, with a little delay. Potassium continues to leave the cell for a short while and the membrane potential becomes more negative, resulting in the hyperpolarizing overshoot. Then the channel closes again and the membrane can return to the resting potential because of the ongoing activity of the non-gated channels and the Na+, K+ pump. All of this takes place within approximately 2 milliseconds, figure 12.24. While an action potential is in progress, another one cannot be initiated. That effect is referred to as the refractory period. There are two phases of the refractory period: the absolute refractory period and the relative refractory period. During the absolute phase, another action potential will not start. This is because of the inactivation gate of the voltage-gated Na+ channel. Once that channel is back to its resting conformation, less than -55 millivolts, a new action potential could be started, but only by a stronger stimulus than the one that initiated the current action potential. This is because of the flow of K+ out of the cell. Because that ion is rushing out, any Na+ that tries to enter will not depolarize the cell, but will only keep the cell from hyperpolarizing. Figure 12.24 stages of an action potential plotting voltage measured across the cell membrane against time. The events of the action potential can be related to specific changes in the membrane voltage. 1. At rest, the membrane voltage is -70 millivolts. 2. The membrane begins to depolarize when an external stimulus is applied. 3. The membrane voltage begins a rapid rise toward +30 millivolts. 4. The membrane voltage starts to return to a negative value. 5. Repolarization continues past the resting membrane voltage, resulting in hyperpolarization. 6. The membrane voltage returns to the resting value shortly after hyperpolarization. Propagation of the action potential. The action potential is initiated at the beginning of the axon, at what is called the initial segment. There is a high density of voltage-gated Na+ channels so that rapid depolarization can take place here. Going down the length of the axon, the action potential is propagated because more voltage-gated Na+ channels are opened as the depolarization spreads. This spreading occurs because Na+ enters through the channel and moves along the inside of the cell membrane. As the Na+ moves, or flows, a short distance along the cell membrane, its positive charge depolarizes a little more of the cell membrane. As that depolarization spreads, new voltage-gated Na+ channels open and more ions rush into the cell, spreading the depolarization a little farther. Because voltage-gated Na+ channels are inactivated at the peak of the depolarization, they cannot be opened again for a brief time, the absolute refractory period. Because of this, depolarization spreading back toward previously opened channels has no effect. The action potential must propagate toward the axon terminals. As a result, the polarity of the neuron is maintained, as mentioned above. Propagation, as described above, applies to unmyelinated axons. When myelination is present, the action potential propagates differently. Sodium ions that enter the cell at the initial segment start to spread along the length of the axon segment, but there are no voltage-gated Na+ channels until the first node of Ranvier. Because there is not constant opening of these channels along the axon segment, the depolarization spreads at an optimal speed. The distance between nodes is the optimal distance to keep the membrane still depolarized above threshold at the next node. As Na+ spreads along the inside of the membrane of the axon segment, the charge starts to dissipate. If the node were any farther down the axon, that depolarization would have fallen off too much for voltage-gated Na+ channels to be activated at the next node of Ranvier. If the nodes were any closer together, the speed of propagation would be slower. Propagation along an unmyelinated axon is referred to as continuous conduction. Along the length of a myelinated axon, it is saltatory conduction. Continuous conduction is slow because there are always voltage-gated Na+ channels opening, and more Na+ is rushing into the cell. Saltatory conduction is faster because the action potential basically jumps from one node to the next. Saltaire equals to leap, and the new influx of Na+ renews the depolarized membrane. Along with the myelination of the axon, the diameter of the axon can influence the speed of conduction. Much as water runs faster in a wide river than in a narrow creek, Na+-based depolarization spreads faster down a wide axon than down a narrow one. This concept is known as resistance and is generally true for electrical wires or plumbing, just as it is true for axons, although the specific conditions are different at the scales of electrons or ions versus water in a river. Potassium concentration glial cells, especially astrocytes, are responsible for maintaining the chemical environment of the CNS tissue. The concentrations of ions in the extracellular fluid are the basis for how the membrane potential is established and changes in electrochemical signaling. If the balance of ions is upset, drastic outcomes are possible. Normally the concentration of K+ is higher inside the neuron than outside. After the repolarizing phase of the action potential, K+ leakage channels and the Na+, K+ pump ensure that the ions return to their original locations. Following a stroke or other ischemic event, extracellular K+ levels are elevated. The astrocytes in the area are equipped to clear excess K+ to aid the pump. But when the level is far out of balance, the effects can be irreversible. Astrocytes can become reactive in cases such as these, which impairs their ability to maintain the local chemical environment. The glial cells enlarge and their processes swell. They lose their K+ buffering ability and the function of the pump is affected, or even reversed. One of the early signs of cell disease is this leaking of sodium ions into the body cells. This sodium-potassium imbalance negatively affects the internal chemistry of cells, preventing them from functioning normally. Visit this site, http://openstaxcollege.org/l/neurolab to see a virtual neurophysiology lab, and to observe electrophysiological processes in the nervous system, where scientists directly measure the electrical signals produced by neurons. Often, the action potentials occur so rapidly that watching a screen to see them occur is not helpful. A speaker is powered by the signals recorded from a neuron and it pops each time the neuron fires an action potential. These action potentials are firing so fast that it sounds like static on the radio. Electrophysiologists can recognize the patterns within that static to understand what is happening. Why is the Leach model used for measuring the electrical activity of neurons instead of using humans? 12.5. Communication between neurons. By the end of this section, you will be able to explain the differences between the types of graded potentials, categorize the major neurotransmitters by chemical type, and affect the electrical changes taking place within a neuron, as described in the previous section, are similar to a light switch being turned on. A stimulus starts the depolarization, but the action potential runs on its own once a threshold has been reached. The question is now, what flips the light switch on? Temporary changes to the cell membrane voltage can result from neurons receiving information from the environment, or from the action of one neuron on another. These special types of potentials influence a neuron and determine whether an action potential will occur or not. Many of these transient signals originate at the synapse. Graded potentials. Local changes in the membrane potential are called graded potentials and are usually associated with the dendrites of a neuron. The amount of change in the membrane potential is determined by the size of the stimulus that causes it. In the example of testing the temperature of the shower, slightly warm water would only initiate a small change in a thermoreceptor, whereas hot water would cause a large amount of change in the membrane potential. Graded potentials can be of two sorts. Either they are depolarizing or hyperpolarizing. Figure 12.25. For a membrane at the resting potential, a graded potential represents a change in that voltage either above -70 mV or below -70 mV. Depolarizing graded potentials are often the result of Na+ or Ca2+ entering the cell. Both of these ions have higher concentrations outside the cell than inside. Because they have a positive charge, they will move into the cell, causing it to become less negative relative to the outside. Hyperpolarizing graded potentials can be caused by K+ leaving the cell or entering the cell. If a positive charge moves out of a cell, the cell becomes more negative. If a negative charge enters the cell, the same thing happens. Figure 12.25. Graded potentials. Graded potentials are temporary changes in the membrane voltage, the characteristics of which depend on the size of the stimulus. Some types of stimuli cause depolarization of the membrane, whereas others cause hyperpolarization. It depends on the specific ion channels that are activated in the cell membrane. Types of graded potentials for the unipolar cells of sensory neurons. Both those with free nerve endings and those within encapsulations, graded potentials develop in the dendrites that influence the generation of an action potential in the axon of the same cell. This is called a generator potential. For other sensory receptor cells, such as taste cells or photoreceptors of the retina, graded potentials in their membranes result in the release of neurotransmitters at synapses with sensory neurons. This is called a receptor potential. A postsynaptic potential, PSP, is the graded potential in the dendrites of a neuron that is receiving synapses from other cells. Postsynaptic potentials can be depolarizing or hyperpolarizing. Depolarization in a postsynaptic potential is called an excitatory postsynaptic potential, EPSP, because it causes the membrane potential to move toward threshold. Hyperpolarization in a postsynaptic potential is an inhibitory postsynaptic potential, IPSP, because it causes the membrane potential to move away from threshold. Summation. All types of graded potentials will result in small changes of either depolarization or hyperpolarization in the voltage of a membrane. These changes can lead to the neuron reaching threshold if the changes add together, or summate. The combined effects of different types of graded potentials are illustrated in Figure 12.26. If the total change in voltage in the membrane is a positive 15 mV, meaning that the membrane depolarizes from -70 mV to -55 mV, then the graded potentials will result in the membrane reaching threshold. For receptor potentials, threshold is not a factor because the change in membrane potential for receptor cells directly causes neurotransmitter release. However, generator potentials can initiate action potentials in the sensory neuron axon, and postsynaptic potentials can initiate an action potential in the axon of other neurons. Graded potentials summate at a specific location at the beginning of the axon to initiate the action potential, namely the initial segment. For sensory neurons, which do not have a cell body between the dendrites and the axon, the initial segment is directly adjacent to the dendritic endings. For all other neurons, the axon hillock is essentially the initial segment of the axon, and it is where summation takes place. These locations have a high density of voltage-gated Na+ channels that initiate the depolarizing phase of the action potential. Summation can be spatial or temporal, meaning it can be the result of multiple graded potentials at different locations on the neuron, or all at the same place but separated in time. Spatial summation is related to associating the activity of multiple inputs to a neuron with each other. Temporal summation is the relationship of multiple action potentials from a single cell resulting in a significant change in the membrane potential. Spatial and temporal summation can act together, as well. Figure 12.26 Postsynaptic potential summation. The result of summation of postsynaptic potentials is the overall change in the membrane potential. At point A, several different excitatory postsynaptic potentials add up to a large depolarization. At point B, a mix of excitatory and inhibitory postsynaptic potentials result in a different end result for the membrane potential. Watch this video. http://openstaxcollege.org/l/summation to learn about summation. The process of converting electrical signals to chemical signals and back requires subtle changes that can result in transient increases or decreases in membrane voltage. To cause a lasting change in the target cell, multiple signals are usually added together, or summated. Does spatial summation have to happen all at once, or can the separate signals arrive on the postsynaptic neuron at slightly different times? Explain your answer. Synapses. There are two types of connections between electrically active cells, chemical synapses and electrical synapses. In a chemical synapse, a chemical signal, namely, a neurotransmitter, is released from one cell and it affects the other cell. In an electrical synapse, there is a direct connection between the two cells so that ions can pass directly from one cell to the next. If one cell is depolarized in an electrical synapse, the joined cell also depolarizes because the ions pass between the cells. Chemical synapses involve the transmission of chemical information from one cell to the next. This section will concentrate on the chemical type of synapse. An example of a chemical synapse is the neuromuscular junction, NMJ, described in the chapter on muscle tissue. In the nervous system, there are many more synapses that are essentially the same as the NMJ. All synapses have common characteristics, which can be summarized in this list. Presynaptic element, neurotransmitter, packaged in vesicles, synaptic cleft, receptor proteins, postsynaptic element, neurotransmitter elimination or reuptake for the NMJ. These characteristics are as follows. The presynaptic element is the motor neuron's axon terminals. The neurotransmitter is acetylcholine. The synaptic cleft is the space between the cells where the neurotransmitter diffuses. The receptor protein is the nicotinic acetylcholine receptor. The postsynaptic element is the sarcolemma of the muscle cell, and the neurotransmitter is eliminated by acetylcholinesterase. Other synapses are similar to this, and the specifics are different, but they all contain the same characteristics. Neurotransmitter release. When an action potential reaches the axon terminals, voltage-gated Ca2+ channels in the membrane of the synaptic end bulb open. The concentration of Ca2+ increases inside the end bulb, and the Ca2+ ion associates with proteins in the outer surface of neurotransmitter vesicles. The Ca2+ facilitates the merging of the vesicle with the presynaptic membrane so that the neurotransmitter is released through exocytosis into the small gap between the cells, known as the synaptic cleft. Once in the synaptic cleft, the neurotransmitter diffuses the short distance to the postsynaptic membrane and can interact with neurotransmitter receptors. Receptors are specific for the neurotransmitter, and the two fit together like a key and lock. One neurotransmitter binds to its receptor and will not bind to receptors for other neurotransmitters, making the binding a specific chemical event. Figure 12.27. Figure 12.27. The synapse. The synapse is a connection between a neuron and its target cell, which is not necessarily a neuron. The presynaptic element is the synaptic end bulb of the axon where Ca2+ enters the bulb to cause vesicle fusion and neurotransmitter release. The neurotransmitter diffuses across the synaptic cleft to bind to its receptor. The neurotransmitter is cleared from the synapse either by enzymatic degradation, neuronal reuptake, or glial reuptake. Neurotransmitter systems. There are several systems of neurotransmitters that are found at various synapses in the nervous system. These groups refer to the chemicals that are the neurotransmitters, and within the groups are specific systems. The first group, which is a neurotransmitter system of its own, is the cholinergic system. It is the system based on acetylcholine. This includes the NMJ as an example of a cholinergic synapse, but cholinergic synapses are found in other parts of the nervous system. They are in the autonomic nervous system, as well as distributed throughout the brain. The cholinergic system has two types of receptors. The nicotinic receptor is found in the NMJ as well as other synapses. There is also an acetylcholine receptor known as the muscarinic receptor. Both of these receptors are named for drugs that interact with the receptor in addition to acetylcholine. Nicotine will bind to the nicotinic receptor and activate it similar to acetylcholine. Muscarine, a product of certain mushrooms, will bind to the muscarinic receptor. However, nicotine will not bind to the muscarinic receptor and muscarine will not bind to the nicotinic receptor. Another group of neurotransmitters are amino acids. This includes glutamate, GLU, GABA, gamma-aminobutyric acid, a derivative of glutamate, and glycine, GLY. These amino acids have an amino group and a carboxyl group in their chemical structures. Glutamate is one of the 20 amino acids that are used to make proteins. Each amino acid neurotransmitter would be part of its own system, namely the glutamatergic, GABAergic, and glycinergic systems. They each have their own receptors and do not interact with each other. Amino acid neurotransmitters are eliminated from the synapse by reuptake, a pump in the cell membrane of the presynaptic element, or sometimes a neighboring glial cell, will clear the amino acid from the synaptic cleft so that it can be recycled, repackaged in vesicles, and released again. Another class of neurotransmitter is the biogenic amine, a group of neurotransmitters that are enzymatically made from amino acids. They have amino groups in them, but no longer have carboxyl groups and are therefore no longer classified as amino acids. Serotonin is made from tryptophan. It is the basis of the serotonergic system, which has its own specific receptors. Serotonin is transported back into the presynaptic cell for repackaging. Other biogenic amines are made from tyrosine, and include dopamine, norepinephrine, and epinephrine. Dopamine is part of its own system, the dopaminergic system, which has dopamine receptors. Dopamine is removed from the synapse by transport proteins in the presynaptic cell membrane. Norepinephrine and epinephrine belong to the adrenergic neurotransmitter system. The two molecules are very similar and bind to the same receptors, which are referred to as alpha and beta receptors. Norepinephrine and epinephrine are also transported back into the presynaptic cell. The chemical epinephrine, EPI equals, on, nephrin, equals kidney, is also known as adrenaline, renal equals, kidney, and norepinephrine is sometimes referred to as noradrenaline. The adrenal gland produces epinephrine and norepinephrine to be released into the bloodstream as hormones. A neuropeptide is a neurotransmitter molecule made up of chains of amino acids connected by peptide bonds. This is what a protein is, but the term protein implies a certain length to the molecule. Some neuropeptides are quite short, such as metenkephalin, which is 5 amino acids long. Others are long, such as beta-endorphin, which is 31 amino acids long. Neuropeptides are often released at synapses in combination with another neurotransmitter, and they often act as hormones in other systems of the body, such as vasoactive intestinal peptide, VIP, or substance P. The effect of a neurotransmitter on the postsynaptic element is entirely dependent on the receptor protein. First, if there is no receptor protein in the membrane of the postsynaptic element, then the neurotransmitter has no effect. The depolarizing or hyperpolarizing effect is also dependent on the receptor. When acetylcholine binds to the nicotinic receptor, the postsynaptic cell is depolarized. This is because the receptor is a cation channel and positively charged Na+ will rush into the cell. However, when acetylcholine binds to the muscarinic receptor, of which there are several variants, it might cause depolarization or hyperpolarization of the target cell. The amino acid neurotransmitters, glutamate, glycine, and GABA, are almost exclusively associated with just one effect. Glutamate is considered an excitatory amino acid, but only because GLU receptors in the adult cause depolarization of the postsynaptic cell. Glycine and GABA are considered inhibitory amino acids, again because their receptors cause hyperpolarization. The biogenic amines have mixed effects. For example, the dopamine receptors that are classified as D1 receptors are excitatory, whereas D2-type receptors are inhibitory. Biogenic amine receptors and neuropeptide receptors can have even more complex effects because some may not directly affect the membrane potential, but rather have an effect on gene transcription or other metabolic processes in the neuron. The characteristics of the various neurotransmitter systems presented in this section are organized in Table 12.3. The important thing to remember about neurotransmitters, and signaling chemicals in general, is that the effect is entirely dependent on the receptor. Neurotransmitters bind to one of two classes of receptors at the cell surface, ionotropic or metabotropic, Figure 12.28. Ionotropic receptors are ligand-gated ion channels, such as the nicotinic receptor for acetylcholine or the glycine receptor. A metabotropic receptor involves a complex of proteins that result in metabolic changes within the cell. The receptor complex includes the transmembrane receptor protein, a G protein, and an effector protein. The neurotransmitter, referred to as the first messenger, binds to the receptor protein on the extracellular surface of the cell, and the intracellular side of the protein initiates activity of the G protein. The G protein is a guanosine triphosphate (GTP) hydrolase that physically moves from the receptor protein to the effector protein to activate the latter. An effector protein is an enzyme that catalyzes the generation of a new molecule, which acts as the intracellular mediator of the signal that binds to the receptor. This intracellular mediator is called the second messenger. Different receptors use different second messengers. Two common examples of second messengers are cyclic adenosine monophosphate, CAMP, and inositol triphosphate, IP3. The enzyme adenylate cyclase, an example of an effector protein, makes CAMP, and phospholipase C is the enzyme that makes IP3. Second messengers, after they are produced by the effector protein, cause metabolic changes within the cell. These changes are most likely the activation of other enzymes in the cell. In neurons, they often modify ion channels, either opening or closing them. These enzymes can also cause changes in the cell, such as the activation of genes in the nucleus, and therefore the increased synthesis of proteins. In neurons, these kinds of changes are often the basis of stronger connections between cells at the synapse and may be the basis of learning and memory. Figure 12.28 Receptor types. An ionotropic receptor is a channel that opens when the neurotransmitter binds to it. B. A metabotropic receptor is a complex that causes metabolic changes in the cell when the neurotransmitter binds to it. 1. After binding, the G protein hydrolyzes GTP and moves to the effector protein, 2. When the G protein contacts the effector protein, a second messenger is generated, such as CAMP3. The second messenger can then go on to cause changes in the neuron, such as opening or closing ion channels, metabolic changes, and changes in gene transcription. Watch this video. http://openstaxcollege.org/l/neurotrans to learn about the release of a neurotransmitter. The action potential reaches the end of the axon, called the axon terminal, and a chemical signal is released to tell the target cell to do something, either to initiate a new action potential, or to suppress that activity. In a very short space, the electrical signal of the action potential is changed into the chemical signal of a neurotransmitter and then back to electrical changes in the target cell membrane. What is the importance of voltage-gated calcium channels in the release of neurotransmitters? Characteristics of neurotransmitter system: cholinergic amino acids, biogenic amines, neuropeptides, neurotransmitters, acetylcholine, glutamate, glycine, GABA, serotonin, 5-HT, dopamine, norepinephrine, epinephrine, metencophalin, beta-endorphin, VIP, substance P, etc. Receptors: nicotinic and muscarinic receptors, GLU receptors, Gli receptors, GABA receptors, 5-HT receptors, D1 and D2 receptors, alpha-adrenergic and beta-adrenergic receptors. Receptors are too numerous to list, but are specific to the peptides. Elimination: degradation by acetylcholinesterase, reuptake by neurons or glia, reuptake by neurons, degradation by enzymes called peptidases. Postsynaptic effect: nicotinic receptor causes depolarization. Muscarinic receptors can cause both depolarization or hyperpolarization depending on the subtype. GLU receptors cause depolarization. Gli and GABA receptors cause hyperpolarization. Depolarization or hyperpolarization depends on the specific receptor. For example, D1 receptors cause depolarization and D2 receptors cause hyperpolarization. Depolarization or hyperpolarization depends on the specific receptor. Table 12.3. Nervous system: the underlying cause of some neurodegenerative diseases, such as Alzheimer's and Parkinson's, appears to be related to proteins, specifically, to proteins behaving badly. One of the strongest theories of what causes Alzheimer's disease is based on the accumulation of beta-amyloid plaques, dense conglomerations of a protein that is not functioning correctly. Parkinson's disease is linked to an increase in a protein known as alpha-synuclein that is toxic to the cells of the substantia nigra nucleus. In the midbrain, for proteins to function correctly, they are dependent on their three-dimensional shape. The linear sequence of amino acids folds into a three-dimensional shape that is based on the interactions between and among those amino acids. When the folding is disturbed, and proteins take on a different shape, they stop functioning correctly. But the disease is not necessarily the result of functional loss of these proteins. Rather, these altered proteins start to accumulate and may become toxic. For example, in Alzheimer's, the hallmark of the disease is the accumulation of these amyloid plaques in the cerebral cortex. The term coined to describe this sort of disease is proteopathy, and it includes other diseases. Creutzfeldt-Jakob disease, the human variant of the prion disease known as mad cow disease in the bovine, also involves the accumulation of amyloid plaques, similar to Alzheimer's. Diseases of other organ systems can fall into this group as well, such as cystic fibrosis or type 2 diabetes. Recognizing the relationship between these diseases has suggested new therapeutic possibilities. Interfering with the accumulation of the proteins, and possibly as early as their original production within the cell, may unlock new ways to alleviate these devastating diseases.